Hi everyone, my seminar topic is immunotechniques. Immunotechniques are mainly used to determine the concentration of antigen or antibody in a sample. Here we are going to discuss about the three immunotechniques: RIA, ELISA, and Western blotting. First one is RIA. The RIA is a radioimmunoassay. This is a technique for determining the concentration of antigen or antibody in a sample. This technique was first developed by S.A. Burson and Rosalind Yalow in 1960. The basic principle involved in RIA is the competitive binding of radio-labeled antigen and unlabeled antigen to a high-affinity antibody. In this technique, we have a microtiter well which is coated with an antibody and two antigens. One is radio-labeled and another one is unlabeled. Coming to the procedure of RIA, first the microtiter well is coated with an antibody, and to this antibody coated microtiter well, radio-labeled antigen is added, and the antigen antibody interaction is allowed to take place, and the free radio-labeled antigen is removed by washing, and after that radioactivity of the well is measured, and that the radioactivity of the well become maximum or at this point. After that, unlabeled antigens are added to the well. Unlabeled antigen then occupy the place of the radio-labeled antigen, and the free radio-labeled antigens are removed by washing the microtiter well. And at this point, radioactivity of the well is measured. Comparing with the first radioactivity measurement, the second one is become lower because of the removal of radio-labeled antigens. If the concentration of radio-labeled antigens in the well is higher, the radioactivity become high, and if the concentration of unlabeled antigen is higher, the radioactivity become lower. The graph representing the relationship between the radioactivity and the concentration of the antigen is given here. If the concentration of the antigen is lower, the radioactivity become higher, and with the increase in the concentration of antigen, radioactivity is lowered. In this technique, mainly the antigen which is labeled with the gamma emitting isotope and the beta emitting isotope is generally used. We have a fixed quantity of antigen for this technique, and the number of epitopes presented by the labeled antigen always exceed the total number of antibody binding site, and the unlabeled antigen bound to the sample mixture will compete with the radio-labeled antigen for the limited supply for the antibody. Even a small amount of unlabeled antigen is added to the assay mixture of labeled antigen, and the antibody will cause a decrease in the amount of radioactive antigen bound, and this decrease is proportional to the amount of unlabeled antigen bound. This is the picture representing the procedure of the radioimmunoassay. In the step 1, radio-labeled antigens are added to this well, and the free radio-labeled antigen is removed by washing. After that, unlabeled antigen of the sample is added to the well, and the radio-labeled antigens are removed by washing, and after that the radioactivity is measured. Applications of RIA includes drug detection from the blood sample, blood bank screening for contaminants, detection of cancer biomarkers, the measurement of hormone level, measurement of disease-associated antigen level. And this technique is mainly used to detect the low concentration of antigen when using antibodies of high affinity. The next technique is ELISA. ELISA is the enzyme-linked immunosorbent assay. The principle of ELISA is similar to that of the RIA. It depends upon the enzyme rather than the radioactive label. Here, in this technique, the study is mainly depend upon the chromogenic substrate. An enzyme conjugated with an antibody reacts with a colorless substrate to generate a colored reaction product. Such a substrate is called the chromogenic substrate. The enzymes included in this ELISA are alkaline phosphatase, horseradish peroxidase, and beta-galactosidase. This technique is mainly used for the qualitative or the quantitative measurement of antibody or antigen. Based on the concentration of antigen or antibody, a standard curve is drawn. A standard curve based on the known concentration of antibody or antigen is prepared, and from which the unknown concentration of a sample can be determined. There are basically 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. First one is direct ELISA. Direct ELISA is one of the simplest ELISA technique here, and there is a microtiter well which is coated with an antigen, and only one antibody is used which is conjugated with an enzyme. In direct ELISA, antigen is immobilized directly on the microtiter well. Add enzyme conjugated antibody that gets bound with the antigen. Then the free antibodies are removed by washing. Then the substrate is added, colored reaction product is formed, and the color is read by the spectrometric technique. And the direct ELISA is used for assessing the antibody affinity and the specificity. Advantages of the direct ELISA include: it is the fast and simplest method. There is no cross reactivity because of the absence of secondary antibody. And the disadvantages include, which is less specific, no signal amplification, low flexibility. Second one is indirect ELISA. Indirect ELISA is mainly used to determine the concentration of antibody. In the indirect ELISA, the microtiter well is coated with an antigen, and to this antigen coated microtiter well, primary antibody is added and allow the antigen antibody reaction to occur. And then the free antibody is washed away. Then secondary antibody which is conjugated with the enzyme is added, and that secondary antibody get bind to the primary antibody. And then the free secondary antibody is removed by washing. Then the substrate for the enzyme is added, and the colored reaction product is formed, and the color is analyzed by the spectrometry. Amount of colored product is measured by specialized spectrophotometric plate readers. This is the picture representing the procedure of indirect ELISA. So here is an antigen coated microtiter well. To this add primary antibody and wash to remove the free primary antibody. Then add secondary antibody which is conjugated with the enzyme, and the substrate is added, and the colored reaction product is formed. Advantages of indirect ELISA include: it is a signal amplification because several secondary antibodies will bind to the primary antibody. High flexibility. Same secondary antibody may be used for several primary antibody, and higher sensitivity when compared to the direct ELISA. And then disadvantages: risk of cross reactivity between the secondary detection antibodies, and the long protocol compared to the direct ELISA. Next one is sandwich ELISA. This technique is mainly used to detect the concentration of antigen in a sample. In this technique, there is an antibody coated microtiter well, and to this antibody coated microtiter well, antigen is added, and then secondary antibody is added. So antigen is sandwiched between the two layers of antibody. Hence, this technique is known as a sandwich ELISA. Antibody is immobilized on a microtiter well. A sample containing antigen is added and allowed to react with the immobilized antibody and washed to remove the free antigens. Then add second enzyme linked antibody which is specific for the different epitope on the antigen. And the free secondary antibodies are removed by washing. Then the substrate is added and color reaction product is measured, and the analysis is done. Advantages of sandwich ELISA: highest sensitivity among the other ELISA types, high flexibility, high specificity. It involves two antibodies detecting the different epitopes on the same antigen. Disadvantages include time consuming, expensive, challenging in getting antibodies specific for the same target antigen. The next one is competitive ELISA. This technique is mainly used for the small molecule. Here, the antibody is first incubated with a sample antigen, and this antigen antibody complexes are then added to the well plate which containing the same antigen. Unbound antigen is removed by washing the plate. The more antigen in the sample, less antibody will be able to bind to the antigen in the well, hence the competition. Because if the antigen in the sample is higher, major part of the antibodies get bind with that antigen, so that there is no antibody to get bind with the antigen on the microtiter well, so there occur a competition among them. And after that, add secondary antibody which is conjugated with the enzyme and which is specific to the primary antibody. Then add substrate and measure the color intensity. Here, higher the sample antigen concentration, weaker the signal. Concentration of antigen in the sample is higher, the antibody get bind with that antigen, so that the antibody cannot get bind with the antigen that is present in the plate. So that if we add the secondary antibody, the color reaction product will be less. The advantages of competitive ELISA include it is suitable for the small antigens, and the disadvantages include require conjugated antigen and it is less specific. This is a picture representing the procedure of competitive ELISA. The first, incubate antibody with the antigen, and this antigen antibody complex is added to the microtiter well which is coated with the same antigen and wash to remove the unbound antibody. Then add the secondary antibody and the substrate is added and color reaction product is measured. Then we are going to discuss about the two modifications of ELISA, chemiluminescence and ELISA. The chemiluminescence is based on the lexogenic substrate. Light produced by the chemiluminescence during certain chemical reactions provide a convenient and highly sensitive alternative absorbance measurement in ELISA. Lexogenic substrate take place of chromogenic substrate. Light produced during the lexogenic reaction may be detected by its ability to expose the photographic film. The quantitative measurement is done using the luminometer. When compared to the chromogenic study, the chemiluminescence study is more sensitive. The next one is ELISpot assay. ELISpot assay is the modification of ELISA. It allows the quantitative determination of number of cells in a population that are producing antibodies specific for a given antigen or an antigen for which one has a specific antibody. The procedure of ELISpot assay include the microtiter well is coated with a specific capture antibody. Cells are added to the plate. Then they are stimulated for secreting their proteins, and these secreted protein get bind to the coating antibody. A specific antibody is added which is specific to the bound protein, and after incubation free cells are removed by washing. The secreted protein molecule is detected by determination of antibody complex. Antibody complex is detected either enzymatically or fluorescently. Each secreting cells appear as a visible spot of color or fluorescence, and which allows the process of quantitative evaluation of secreting cells. Applications of ELISA. The ELISA technique is used to determine the presence of antibodies and antigen in a sample in the food industry for detecting the presence of food allergen, and in the vaccine development during the disease outbreak, tracking the spread of disease and measuring the hormone level as a test for the pregnancy. The next immunotechnique is Western Western blotting is mainly used to identify the specific protein in a complex mixture of protein. A protein mixture is electrophoretically separated on the SDS page. The procedure includes first our protein of interest is extracted. After that, gel electrophoresis is undergone. The sample is located in the this sample is loaded in the well of SDS page and the proteins are separated on the basis of the charge. Next step is blotting. The nitrocellulose membrane is placed on this gel and this separated protein get transferred into that nitrocellulose paper by capillary action. The next step is blocking. The antibodies are the protein, so they are likely to get bind with a nitrocellulose paper, and before adding the primary antibody, the membrane is non-specifically saturated or masked by using a casein or BC, and this procedure is known as blocking. After the blocking, the primary antibody to the desired protein is added, so antigen-antibody complex is formed. Then secondary antibody, which is enzyme conjugated, is added, and then the substrate is added and colored reaction product is formed. The protein bands are transferred to a nitrocellulose membrane by the electrophoresis. The individual protein bands are identified by flooding the membrane with a radiolabeled or enzyme-linked antibody specific for the protein of interest. Antigen-antibody complexes that form on the band are determined by the autoradiography. This is a picture representing the Western blotting procedure. Applications of Western blotting. This technique is used to identify the presence of specific single protein in a mixture of complex protein and used in the medical diagnostics, that is HIV test, and to determine the size and the amount of proteins in the given sample and useful to detect the defective protein. So here we are discussed about three immunotechniques: radioimmunoassay, ELISA, and the Western blotting.